Okay, hey, we've been talking about phases of mitosis. If you look at these phases as they're represented here, those should be pretty similar to the ones that we've talked about. Only difference is they have this extra phase in here, this pro-metaphase. Um, why am I showing you this graphic? I want to show you this graphic for the uh, graph, image, table, whatever, whatever chart on the top, as that gives us a really good idea at how long each cell spends in each stage. Now, this is going to be different if you're a skin cell versus if you're a stomach cell or a nerve cell, those kind of things. But I like this because it's a general representation of time spent in each stage. If you notice this green, huge outside arc, that is interphase. All right, so for the typical cell, it's going to spend, 95 to 99% of its life in interphase. And that should make sense to us. That's the cell doing its cell stuff. That's the stomach cell being a stomach cell. That's the skin cell being the skin cell. It's not worried about dividing. It's doing its own things. If you notice, more advanced, we break down interphase into a couple different things that are going on here, including some copying of chromosomes at the end. We don't spend a ton of time on that, but just knowing that proportionally the cell spent its life in interphase, and then when it goes to divide, it goes through this quick mitosis phase in there. All right. So inside of this process, we want to talk about how it gets controlled. Okay, so what causes things to divide, what causes things to grow. We'll talk a lot tomorrow about the implications of this, and well, just when you grow new cells, usually that could be actual growth of a body. It could be healing of different body parts, etc. And so it's curious to us. So the eukaryotic cell is regulated by a molecular control system. So that interphase that we talked about is divided into three separate portions. We saw this on the other diagram: G1, S, and G2. So G1 is that initial growth. So it's a small cell. It grows to fulfill its required size. It goes through the S phase where the chromosomes are duplicated. And then G2, it finishes growing and prepares for cell division. All right. So those are three stages within interphase itself that are crucial for that cell to be ready. Because if it goes through mitosis and then it starts dividing again right away, that actually causes a lot of problems that we're going to be exploring. In these stages, there are some really strict checkpoints. So your body regulates itself very well. We don't want any errors in any copying. So a checkpoint's a control point where either stop or go ahead signals can regulate cell reproduction. So if it isn't meeting the requirements, it might the cell is programmed to die or to fix itself, etc. So once again, G1 checkpoint, the restriction point, that's the most important one. If it passes the check, the cell will head towards division. If it fails, it enters this G sub 0 phase. So G naught, a non-dividing cell is what it turns into. So a good chunk of cells in your body are done dividing. All right. They just are. When it comes time, and this is more of an anatomy deal. I talk about like your skin cells. Well, there's a certain stretch of skin cells that make new copies, but the skin cells themselves are kind of their own little individual appeal there. Same with muscle cells, nerve cells, cartilage cells. You have some specialized stem cells throughout your body, and that's different topic for a different day. But in an emergency, a lot of them can be called out of that G sub 0 phase and do mitosis there. So like I said, here's just a little more breakdown where we have G sub 1, synthesis, G sub 2, and then if it is a cell that is, hey, it's just going through the resting period and just going to do its job the rest of the time, it's going to enter this G sub 0 phase. So I like that diagram a lot. That's a really helpful one. Take a screenshot of that, save that picture. That's a nice reference point to have there. All right. And then when I say it's controlled, how is it controlled? There's a couple enzymes that regulate the cell cycle. Two that we're going to write down are protein kinases and cyclins. Like I said, they're just little chemical checkpoints, like spell checkers, to make sure that everything's being copied correctly, that you have full samples of everything. And then I don't know if we've mentioned what enzyme is before or not, but enzymes are proteins found throughout the body, and they catalyze a variety of reactions there. Okay, so a little bit kind of wrapping up phases of mitosis and how much time we spend in each, and how those things work out together.